Welcome everyone to Cannabis 101, what you need to know to answer your patient's questions and feel good about it. To give you a little history on where I come from, in 2013, I thought cannabis was a gateway drug. In 2014, after being requested to step into the space and problem solve some kids that were having intractable side effects from chemotherapy, I investigated cannabis, and by the end of 2014, I was already working on a patent. Here's my disclosures. I am involved in both research and drug development in the cannabis industry today. So our learning objectives for this presentation, I'd like to give you an overview of cannabis and its constituents and review some of the most important families in our phase 1 or cytochrome P450 elimination system impacted by phytocannabinoids, review the different delivery methods of cannabis and their impact on onset and duration of effects, and review common side effects of cannabis and how they relate to route of administration. Now, you all should have access to the slides and references so that you can go back and review details that you find interesting. My goal is to expose you to important information and challenge you as healthcare providers to investigate this very powerful and safe adaptogenic herb. Now, I think cannabis is a beautiful plant. On the right, you're looking at a cannabis leaf, and on the left, you're looking at what's called a bud, or a cured, trimmed form of the cannabis flower, which is in its prime consumable form. Now you're looking at a couple pictures, one on the left looking at Santa Cruz Tsunami, a 4-to-1 ratio marijuana plant, and to the right you're looking at a hemp cannabis plant that has less than 0.3% THC in it. To the untrained or trained eye, today, it's very difficult to tell the difference between different forms of cannabis, that being marijuana or hemp. So let's take a look at what those terms really mean. Both are cannabis sativa L, but they're regulatory terms, and it's based on the content of THC. Marijuana being defined as containing greater than 0.3 milligrams per gram of THC in the dry weight of the plant. And remember that still today, federally, marijuana is a Schedule I drug, and by definition, that means there is no recognized medicinal value and it's highly addictive. Hemp, on the other hand, is defined as having 0.3 milligrams per gram or less of THC in the dry weight of the plant. And in the amended Farm Bill of October of 2018, Congress defined hemp finally as an agricultural commodity, both the plant and all of its components. And in states that have defined a USDA program which makes hemp growing legal in that state, by definition, if it's legal in the state, then it's federally legal by default. Now, that was October of 2018. We're in March of 2020, and the federal government still has not rolled out their federal program. Now, Cultivar versus chemovar versus strain. Plants do not have strains, bacteria and fungi do. But strain is a common term that is talked about, especially in dispensaries. Cultivar, which is an equivalent and proper term to strain, is generally defined by the grower based on smell, appearance, and physiological effects. Now, common terms used are sativa, indica, and hybrid. Sativa, typically a taller, skinnier plant, and the physiologic effects are described as more energetic and giving someone a head high if they're consuming THC. Indica is a shorter, squatty plant, broader leaves, and generally gives an effect of being more relaxing and more of a body high. Now, greater than 95% of the flower market today is all hybrid or mixes of sativa and indica. And there's a few varietal names that you may have heard: Jack Herrera, sativa dominant, blueberry, and indica dominant, and catatonic hybrid. Chemovar, on the other hand, is a term that is defined by multiple chemical components which can be standardized, identified, and most importantly, will need to be used in order to ensure reproducible cannabis effects, physiologic effects that patient can expect. Now, the endocannabinoid system, we could spend 2 hours talking about. I've only got a few slides, and so this is an area that I implore all of you to investigate. The endocannabinoid system regulates physiological and cognitive processes and is probably the most important physiologic system establishing and maintaining health and homeostasis. It's involved in fertility, pregnancy, prenatal and postnatal development, appetite, pain cessation, mood, memory, neuroprotection, inflammation. Endocannabinoids play an important role in the neurotransmitter, in the immune system, mitochondrial function, and they have anti-proliferative, anti-inflammatory, and anti-metastatic effects. And back in the 80s, when I was in medical school, we were told that the runner's high was from endorphins, but later we have found that it's actually in our endocannabinoid system and anandamide, one of the endocannabinoids. Now, endocannabinoids are endogenous lipid-based retrograde neurotransmitters. Anandamide and 2-AG for short are two of those endocannabinoids. THC mimics anandamide, and CBD mimics 2-AG. So this is, in our endocannabinoid system has kind of a key and a lock system. The receptors that were first discovered, CB1 and CB2, are G protein coupled receptors that are on the plasma membrane of each cell in our body. CB1 is mainly found in the central nervous system, although scattered throughout the body. CB2 is mainly in the peripheral system, but is also found centrally. CB1 receptors were first cloned in 1990, and CB2 receptors were first cloned in 1993. Now, we have cannabinoid receptor dependent and cannabinoid receptor independent mechanisms that endocannabinoids as well as phytocannabinoids engage in. And this is a list of some of those receptors, and you can review on your own leisure. So let's look at the phytoconstituents that come out of the cannabis sativa plant. They all start as an acid form, and the main acid that all other cannabinoids are derived from is CBG. This chart shows you the enzymes also involved in generating the main phytocannabinoids. Now, when the cannabis flower is harvested, it contains mostly acid, but temperature and light will help the decarboxylation or the conversion of the acid form of that cannabinoid to the neutral form. We're looking at the two main cannabinoids on the left, THC, and on the right, CBD. Notice that these acid form and neutral form have physiologically active value. And at the bottom, one of the terpenes, beta-caryophyllene, is a direct CB2 agonist and has its own direct anti-inflammatory activity. So again, we're taking a look at the two main phytocannabinoids, THC and CBD. THC being a partial agonist of both the CB1 and CB2 receptors. Activation of the CB1 receptor centrally is where we get the effect of the intoxicating effect, or that high that's associated with THC in the neutral form, not in the acidic form. And reported effects are listed there. CBD is an allosteric modulator of CB1 and CB2 receptors. It's an inhibitor of FAAH and an agonist of the serotonin and phenylonoid receptors. Now, Miri's work out of Israel, he's done tremendous work. This is looking at a heat map, an LCMS approach, and I apologize for the size of this, but on the top of this heat map lists 36 cultivars, and on the left side, it's identifying the different cannabinoids. Now, there's been 144 cannabinoids identified to date, and over 500 physiologically active substances in the cannabis plant. So when we simply talk about THC or CBD, it's remarkable that you can see, dark areas are high concentrations, light areas are low concentrations. There is 10 different THC delta 9 types. There is 10 CBD types just identified in a portion of this heat map. Looking at terpenes, which are the aromatic substances that give plants their unique scent and flavor, there's three categories: mono, di, and sequesterpenes. Most cannabis plants can possess up to 200 terpenes, but there's typically 5 to 8 dominant terpenes. Terpenes possess their own individual effects and many times dominate the subjective experience of a cannabis user. Now, Leafly.com is a good internet source that has tried to standardize some information both with color and with category. And so this is one of the ways that they present how, for this example, white Tahoe cookie, it's an indica dominant cultivar, and it's showing the dominant terpenes that are color-coded, and you can find those colors associated with a calming effect, myrcene being on the far left, and energizing effect with terpenoline on the right. Here is an information wheel that they've created that looks at terpenes, boiling points, aromas, the effects found, some of the medical benefits that are associated with different varietals. And so the colors, green being hybrid, red being sativa dominant, and purple on the outside of the circle being indica dominant. This is where patients are going to be coming to you with questions, and this is some of the resources that they've been looking at. So that's why I'd like you to become familiar. So looking at a basic single dose pharmacokinetic curve, Cmax separates the absorption from the elimination phase. The therapeutic range is defined between the minimum effective concentration and the minimum toxic concentration. Tmax is the time it takes to reach Cmax, and the time to onset is defined as reaching the minimum effective concentration. The therapeutic range defines efficacy. Cannabis pharmacokinetics fit into this model, but many of the therapeutic endpoints do not because they are biphasic. Inhalation is the most common and preferred route to date, and is also the most published route of administration in the literature. Now, bioavailability of THC depends on the way of smoking, and there is a number of different ways to inhale. And what's remarkable is even with control of both content, temperature, inhalation, volume, length of time breath held, you still see between a 6 and 12-fold difference. If you look on the far right-hand side, THC range, 12-fold difference from one individual to the next with the same controlled inhalation method of any given dose of cannabis. This is pretty profound, and why it makes it so difficult to know exactly what someone is going to experience. And many of these influences are genomically based. So this is controlled inhalation, and clearly this is an example of not a controlled inhalation method. Typically, 100 nanograms per mil is considered the concentration of THC in the blood that causes intoxication. But again, this is really dependent on the individual and their tolerance. Sorry about that. So let's quickly look at the first and a number of the FDA-approved cannabinoids that Marinol was the first being FDA approved back in 1985. So there is actually a lot of data on, synthetic, starting with dronabinol, synthetic THC. This is a soft gel capsule that's swallowed. What's interesting is you have an 90 to 95% absorption rate, but only 10 to 20% gets into the bloodstream. Here's another formulation of dronabinol, which is an oral solution in an ethanol propylene glycol base. Taking a look at the first synthetic THC analog, which is nabilone, which is consumed orally as a capsule, having very similar bioavailability. The first phytocannabinoid to hit the world market by GW Pharma was Sativex, or nabiximols. This is a approximately 1-to-1 ratio of THC to CBD, and it is delivered as an oromucosal spray. So this gives us some good pharmacokinetic data on both THC and CBD. It's Sativex is currently still in phase 3 clinical studies here in the U.S., but it is currently available in at least 36 countries that have already approved it through their FDA equivalent federal drug approval process. In June of 2018, the first phytocannabinoid extract from marijuana, Epidiolex, another GW Pharma's product, was approved for the use in seizure or treatment-resistant seizure disorders. Again, this is a CBD product. It is marijuana-derived, a phyto extraction, and is not a synthetic. Now, to appreciate the difference in formulation of the same drug, take a look at THC detection in plasma after 15 minutes comparing Marinol and Syndrose. Marinol being swallowed as a capsule and Syndrose being a liquid. So besides at 15 minutes, less than 25% of Marinol is being detected in the plasma, where 100% of Syndrose is. There's also a substantial intra-individual variability between Marinol and Syndrose, if you look down at the bottom line. Now, looking at the pharmacokinetics of terpenes, Kohlert in 2000 did a great review of and looked at the published data in both humans and animals. There are more than 3,000 compounds that have been described, but limited studies on most. On the clinical side, there's been a lot of published data on essential oils and well-established in vivo studies looking at COPD, acute bronchitis, IBS, and the rest. Pharmacokinetic data, though, on terpenes and terpenoids is very limited. And really the bottom line is that the analytical methodology is the rate-limiting step to the quality and reproducibility of data. So in order for us to really appreciate the value of terpenes therapeutically in cannabis, we're going to have to do a much better job of establishing standards in analytical methodology. The main drug interactions involved with cannabis involve the phase 1 elimination system or the cytochrome P450 system. More than 60% of prescription medications are metabolized by this phase 1 elimination system in our liver. All phytocannabinoids inhibit certain CYP450 enzymes and involve mainly 3 families, the CYP2C, 2D, and 3A families. CBD is the strongest inhibitor of all of the phytocannabinoids studied to date. Now, what's important to realize is most studies show little clinical impact when using normal therapeutic dosing of cannabinoids. Many of the studies that are looking at the theoretical impact use 10 to 400-fold higher concentrations that are found clinically. Now, there's a few drugs that clearly have an impact, drug-drug interaction with cannabis, and it's important to identify those and know how to work with that. Clearly, warfarin is one of those drugs. So in this study, looking at 85, this was a review article looking at 85 articles. There was 3 eligible articles that were identified back in January of 19. One looking at the in vitro effect of THC and its inhibitory effect on CYP2C9 and warfarin, which is one of the metabolic pathways for warfarin, and 2 in vivo. One, a male consuming marijuana, weren't sure about the dosing, but there was 2 situations where his INR was raised above 10, and there was some associated bleeding. One of the medications for epilepsy, Epidiolex, required a 30% reduction in warfarin in order to maintain that therapeutic INR value. Now, what's frustrating is the authors suggested that if the patient was on warfarin, they should be warned against cannabis use. We warn patients about the use of dark green leafy vegetables. We tell them if you're going to consume them, which many of them contain the antidote to warfarin, which is vitamin K, we just say stabilize your diet. There is no reason why a patient can't be on warfarin and cannabis as long as that adverse event is being managed properly by following INRs. Now, another significant drug interaction with cannabis is with clobazam, which is a anti-seizure medication for treatment-resistant cases. And clobazam and its active metabolite, norclobazam, can have a profound impact on or blood levels. And so if cannabidiol or CBD is being used, it's very important to monitor both clobazam and norclobazam at 4 and 8 weeks of starting therapy. And this study, in a great randomized crossover study, they looked at 3 different drugs, looking at those different pathways that are known to be inhibited mainly by cannabinoids. The 3A4, the 2C19 are main areas, and looking at rifampin, an inducer, ketoconazole, an inhibitor of 3A4, and opramazole, a PPI, that is an inhibitor of the 2C19 pathway. What they found is that if there is a patient on cannabis and on another prescription medication that is influencing either by inhibitory or inducing the 3A4 pathway, you should take a look at that impact in potential drug-drug interaction. Project CBD is a fabulous reference for healthcare providers and patients. I strongly recommend that you visit this site and learn the science as well as the real-life stuff involved with cannabis therapeutics. It's also available besides English and Spanish, Portuguese, and Japanese. So let's look at the adverse effects using cannabis. This is a partial list. If you look closely, that you'll find that many of the reported adverse events from cannabis are actually treatment endpoints, or for instance, insomnia is one treatment endpoint that cannabis is used for all the time. Anxiety, panic attacks, very valuable in these areas. Factors that influence these adverse events include the formulation, route of administration, the experience and tolerance of the patient, any comorbid conditions that they have, drug-drug interactions, the genomics of the individual, whether there's product contamination, and whether the products have been misbranded or mislabeled. So knowing what to use and how to use it is the key to avoiding almost all adverse events. Now, Dr. Marcoux is going to be talking about inhalation and vaporization, and so I'm not going to spend much time on this, but I did want to just make you aware of some of the key points surrounding vaporization hazards. One of them is temperature and its influence on breaking down, for instance, terpenes in this particular study, and where myrcene, limonene, and linalool will break down under certain temperatures. It can form isoprene and then ultimately some very toxic byproducts like benzene. Temperature is important, and what the experts say is it should, for dabbing or combustion, it should never go above 378 degrees Celsius. Now, recently, there's been a huge amount of concern over e-cigarettes, and it seemed like this just all of a sudden started popping up in 2018 and 2019. But this data has been available as early as 2017, looking at the carbonyl compounds produced from vaporizing cannabis oil using thinning agents. We should never see a thinning agent, a flavoring agent, ever being used in a cannabis product, cannabis oil that's being vaporized or being used in or added to flour. What we find is that the products that have been studied can produce some pretty toxic things. Polyethylene glycol, for instance, vitamin E acetate, even MCT, medium chain triglycerides, polypropylene glycol, can produce some pretty nasty stuff. PG and PEG can produce acetaldehyde and formaldehyde, which there is no safety, exposure to those toxic products. And PEG is most important in producing other carcinogens like 1,4-dioxane and ethylene oxide when it's heated. So bottom line is never smoke an e-cigarette, don't use flavoring agents to inhale, and don't ever expose yourself to a cannabis extract that's vaporized. And what's really good about regulated markets today is that none of the products that you find in a dispensary today, unlike what's found in the black market, have diluents that can cause these problems. Now, looking at the disconnect between patient, physician, and cannabis, regulatory framework surrounding Schedule I drugs has always separated the patient from the physician. And that is because if a physician prescribes a Schedule I drug like cannabis, that is aiding and abetting a federal felony. And that is one of the problems that we're still dealing with physicians having a DEA registration and operate in a state that allows for both medical and or adult use consumption. So what, I'm giving you an example of Nevada, but every state should have statutes that protect a licensed healthcare professional from both their licensing boards for any disciplinary action, and I implore you to take a look at these areas. There's always a qualifying condition when there's medical and or, medical use. Adult use doesn't require a qualifying condition. And there's different qualifying conditions based on each state. There's generally a process that a medical patient needs to follow in order to get the ID necessary. When medical patients, unlike adult use patients, get registered, that registration generally allows them to avoid taxes, and it also gives them access to products that are much more concentrated or in a larger quantity per package than an adult use patient can get access to. In each state, there is generally regulations associated with possession and consumption. And typically medical patients will have access and be able to carry more product on them at any one time or purchase more product than adult use. And generally there is rules for consumption. And so you need to know what those state and county rules are, because they vary between state and between county and sometimes even city. Now, the National Academies of Sciences, Engineering, and Medicine, 18 years after their first report, did an extensive review of the literature, January of 2017. Actually, that's when it was published. It looked at over 10,000 research articles, all human based. So they removed all the in vitro. Let's start again.
No worries. This is 50 still, right?
Ready to start. Yep.
Okay. All right. I'll count you down. 5, 4, 3, 2.
Now, the National Academies of Sciences, Engineering, and Medicine, 18 years after their first report, did an extensive review of the literature in January of 2018. And looking at only human data in over 10,000 research articles, came up with 100 research conclusions. From this information, they defined level of evidence as conclusive, substantial, moderate, limited, or no or insufficient evidence. What is remarkable is that they found conclusive evidence for the use of cannabis in nausea and vomiting associated with cancer chemotherapy, in chronic pain in adults, and spasticity in MS. Chronic pain in adults. We've been in an opiate crisis for years now, and still the federal government doesn't recognize cannabis as having a valuable place in medicine still today with their continued Schedule I category. This in 2019 looked at over 800,000 registered patients in 20 states, including Washington, D.C., and found that 85.5% of the conditions for which the patients are licensed to use medical cannabis either have substantial or conclusive evidence of efficacy. Here's another information wheel from Leafly that lists symptoms and conditions. And again, this is a resource that many patients may even bring into your office and ask you questions about. Now, the reported medical applications of cannabis are broad. This is just a partial list. In our clinic in Huntington Beach, we find greater than 60% of patients are looking for improved pain control and or to get off of their opiates. Anxiety is another very high indication for the use of cannabis, as well as many of the other indications that have been discussed earlier. In this study of autism, children with autism spectrum disorder, what is remarkable to me, besides this being a great resource for references, is that in this 53 children group, the side effect or adverse event profile of somnolence or a change in appetite were all mild, but to look at self-injury and rage, hyperactivity, sleep, a profound problem with autism spectrum disorder, and just overall improvement. These are high 60 to low 70% improvements in the use of cannabidiol in this patient population. Profound. Routes of administration. Now, there's different ways to use cannabis, and you can think of almost any pharmaceutical. There is a product that has cannabis in it, it's available. So whether it's vaporization, combustion, smoking like a joint, edible forms, capsules, sublingual tinctures, buccal absorption. We'll go over some of these and the importance of why all of these different forms should be always made available to a medical patient. Vaporization, or the combustion of plant material, is still the most frequently used form of administration of cannabis today. Generally, we look at a quick onset within 1 to 5 minutes and a duration which is short, generally 1 to 2 hours. Again, it depends on the device. Some devices, like the Dosit to the right here, can deliver a specific dosage of cannabinoids that are defined and allow a patient to know exactly, as well as the healthcare provider, to know exactly what the patient is consuming and the dose in order to see what that patient's outcome is. Now, there's different standardized delivery systems that are out there around the country and the world that help deliver very specific doses of cannabinoids by inhalation. This is one study that looked at 6 patients, and the important thing to look at is that using the Volcano device, which controls dose, volume of inhalation, you still see a Cmax of 30 to 180 nanograms per mil. This is the same dose, the same inhalation volume, and looking at 6 different patients and having that variability in the amount of cannabinoids measured. This is a study looking at over almost 3,000 patients, all above 65 years old, mean average age of 74.5 with pain and cancer. They looked at after 6 months of cannabis use, how the respondents improved in their condition, which is profound. Their study found that therapeutic use of cannabis is safe and efficacious in the elderly population. It may decrease the use of prescription medications, including opioids. I urge you to take a look at that study. Now, there's edible forms of marijuana. Many states will define different edible forms statutorily differently from state to state. But generally, edibles should be considered a food product. So cannabis has been infused into something that you eat. Onset of action can be 1 to 2 hours, up to even 4 hours, depending on what the patient ate before they consumed the cannabis. And if you've eaten a heavy, fatty meal, it could take 4 hours for gastric emptying to occur. Duration of action is generally 6 to 8 hours. Edibles are where you hear the most extreme experiences, where consuming a brownie, for instance, waiting an hour, no effect, eating another serving of a brownie, still no effect, and then 4 hours later, the person is so high that they're very uncomfortable and intoxicated. So it's, again, a much more difficult route of administration to control. Here's a study that you can look at, looking at a 10, 25, and 50 milligram dose of THC and the blood levels that were observed in both the metabolites and looking at THC. And it's clearly not linear. Tinctures, sublinguals, buccal absorption. This typically is very dependent on formulation and placement in the mouth. So buccal is between lip and gum. Sublingual is typically placed underneath the tongue. And the problem with assuming that one product, a tincture or a sublingual solution, is equivalent to something else that possibly is delivered buccally, you can't make an assumption unless you know the specifics about the formulation that the patient is taking. So if one person has, not a good outcome with the sublingual, don't assume that all of those products out there are going to be the same. Depends on how they administered it, whether they held it in their mouth for 5 to 15 minutes before swallowing. If you put a sublingual in your mouth and you swallow it within 20, 30 seconds, which most patients do because they don't like the taste, then all of a sudden you've turned oral mucosal absorption into an edible. And as such, you're going to get a much slower onset of action and a different duration of action. Most sublingual products on the market in the cannabis space are sublingual oils, and they have a 15 to 45 minute onset of action in a duration of 3 to 4 hours. Here's some examples of a couple published products, tablets, or a fast melt tab. Here's an example of misbranded products. In a pharmacy, you would not find a sublingual solution like Drift Away labeled as a tincture. Tinctures are typically an ethanol-based solution and not a coconut oil-based solution. Here's a quick study looking at the impact of minor cannabinoids. This patient had relatively intractable seizures, treatment-resistant seizures, at the point that he was seen and given a THC CBD sublingual solution with no terpenes based in MCT oil. He was having 8 seizures a day. Within 24 hours, he had seizure-free days that continued until he went back to get another refill from his local dispensary of what he thought was the same 3-to-1 sublingual solution. And what occurred was immediate loss of seizure control and seizures starting again. So the dispensary staff member that helped this patient was also aware that they changed the cultivar from a Diamond OG to a lemon haze in that particular product. He went back, made more of the Diamond OG extract, and again, the patient regained seizure control. And if you look, there's in the phytocannabinoids that we are looking at, which may or may not have a causal effect, CBGA and CBC were really the only significant difference between those. Transdermal patches and creams, there's just tons of products out there. The majority of them do not produce a psychoactive effect if it contains THC in the neutral form. There is CB1 and CB2 receptors all over the surface of our body, and many people do get great relief from the topical application of products. It's important to know or follow a few guidelines for patients new to cannabis. You always want to start low, go up slow. 1 to 5 milligrams of cannabinoids are typically considered a microdose. If the patient has never consumed cannabis before, generally after a single dose of a microdose, you want to wait 3 hours before consuming more, especially if it is an edible, like the gummies that you're seeing to the right. Remember that CBD is an antidote to the THC intoxicating effects. In our clinic, we never start a cannabis-naive patient on any THC-containing cannabinoids. It's always started, we always start them on a CBD product between 1 and 3 days, see how that impacts their symptoms, and then add THC at night, generally in a 1.25 to 2.5 milligram dose. Remember, start slow, start low and go up slow. You can always add more, but it's hard to take away what you've consumed that is causing undesirable intoxicating effects. Laboratory testing is critically important, and all states that have medical and or adult use require testing. And we're looking at potency and any risks associated with pesticides, residual solvents, heavy metals, and microbiological or mycotoxin contamination. This is an example of some of the equipment that's used. LCM SMS is really a great way to have analysis. And this is an example of a certificate of analysis. This is in reference to an LA-AFI flower that is being tested out of Nevada. So you should never buy a product, whether it's in a dispensary or it's a CBD product on the market, without a certificate of analysis for every batch and product that you're considering to buy. If it doesn't come with that, don't buy it. So something to think about. Cannabis is an adaptogenic herb, and it's disease state modifying. Adaptogens are substances that help you arrive at homeostasis. Cannabis has a huge therapeutic range and no LD50. Cannabis is a federally Scheduled I drug still today. Politics and special interest, not evidence-based medicine, results in its prohibition and Schedule I drug status. The endocannabinoid system is profound, and it drives our homeostasis. Cannabis has been used for thousands of years in multiple cultures. Cannabis was one of the top 3 prescription drugs prescribed by physicians in the US before 1932. It was in the Pharmacopoeia from 1850 to 1942. So my recommendation is educate, embrace, and empower. And with that, I leave you with Dr. Burgess and Dr. Crowley in the field of legal hemp.